In this video, we'll be going over the answer key for Monday, April 20th. And what we're talking about here is still enzymes, so we have a little review based on what we talked about on Friday. And this is a review in general chemistry, just to see if you remember what type of reactions happen in chemistry, and then pair them up with the enzyme that would catalyze that reaction. So in A, we have a CH3 group and a chiral carbon, and we're changing the chirality of this carbon by having this CH3 group move on to the other side. That is just an isomerization reaction. We're not changing any of the molecules, we're just kind of changing how they're bonded. B, we have a COO minus up here, and we're chopping that off to form C carbon dioxide, O double bond C double bond O. We are eliminating a group, so that would be a lyase. Here we have a molecule, and we're adding NADH, and we're taking this C double bond O, and we are reducing it to OH. So anytime you see NADH or FADH2, that's going to be a redox reaction, and that would be an oxidoreductase. D, we have ATP, and we have NH4, and what we're doing is that we're taking this NH4, attaching it to our carbon, and we're losing this oxygen. So here it's a bond formation because we're forming a new bond and we're using ATP. So that would be a ligase. Alright, so today we're going to focus on some mechanisms. And we're going to start with mechanisms of acid and base chemistry by looking at this ketol to enol transformation. And basically during this transformation, one of the hydrogens from this CH3 group leaves and you form a double bond between this carbon CH2 and this oxygen gets a hydrogen. Hydrogens on carbons aren't acidic, so this is an extremely slow reaction. So we can catalyze this by either doing an acid or base catalysis. In the acid catalysis, we will present an acidic hydrogen to this oxygen. That oxygen will try and take this hydrogen, which would make this carbon partially charged. Carbons hate to be charged, and so to fix that, one of the hydrogens on this CH2 group would want to leave. And again, this carbon now would be partially charged. That carbon doesn't want to do that, so you'll form a double bond while this oxygen will get a hydrogen. And for enzymes, they need to be replenished or reformed after the reaction is done. Otherwise, they would be used once and then more or less be dead. So we need to give this acid molecule another hydrogen. So that can come from water. It's usually how it happens inside the cell. Here we have a base catalysis. Similar idea, except this time we have a base that will want to take the hydrogen away from this CH3 group. And when it's taking the hydrogen away, you get a partially charged carbon. So you want to form a double bond. If you need to form a double bond between these two carbons, you can't form a double bond with this oxygen, which makes it negative. Water can come in and give a hydrogen away, and then you have your enol. Another mechanism we're going to look at that's acid and base is RNA Ace. RNA Ace cuts RNA molecules and does so by histidines. We have one histidine that acts as a base, that would be histidine 12, and one that acts like an acid, that's histidine 19. So our first mechanism, our first reaction in this mechanism is 12, takes a hydrogen from our 2' hydroxyl. Again, this only happens in RNA. If this was DNA, this would just be carbon hydrogen. When we take that oxygen away, the oxygen will be negatively charged, so it'll attack this phosphate. And to help split this RNA molecule, we have another histidine that's going to act as an acid to give the hydrogen to this oxygen, breaking this phosphate oxygen bond and forming a new oxygen phosphate bond. So you'll get a cyclic nucleic acid. And you'll also have water come in after the first step. Now, we don't want this cyclic nucleic acid to stick around. We also need to rechange our histidines to how they were like at the beginning of this mechanism. So our hist-19 is going to take a hydrogen from water. This OH will attack this phosphate. Then our RNA will take this oxygen from this hydrogen, and you will get an RNA out that's no longer cyclic, and your histidines are in the correct state so that they can take in another RNA and chop it up. So question 2, looking at your acid and base catalyzed reactions, chemically speaking, how are they speeding up this reaction? And they both come down to the same answer fundamentally. We are stabilizing the transition state. They do it in different ways, but at the end of the day, we're making this transition state easier to get to. And if you remember what we talked about last week, the speed of the reaction is solely dependent on the height difference or the energy difference from the reactants to the transition state. That gap is your reaction speed. So in the acid, we're presenting a hydrogen to this carbonyl group, which wants to take the hydrogen away, making this hydrogen on the CH3 more likely to leave. On the base, we are trying to take away this hydrogen from the CH3, which will allow the double bond formation to form easier. But yeah, at the end of the day, it's really stabilizing that transition state. C, both His can act as an acid and base. Why is His good for this? Because the pKa of histidine is roughly 6. And if you remember, was with pKa, that's telling you when you're going to pick up an acid or a hydrogen, when you're going to drop off a hydrogen. So since it's 6, you can actually change the pKa based on its local environment. So you can make one hydrogen pKa is higher than 6, one lower than 6. But what this ultimately means is that at the pH of the cell, which is roughly 7.2, histidine can act as an acid or a base because of its pKa being at 6. Question 3, going back to our mechanism, why can't RNA cut DNA? It needs that 2' OH group, that hydroxyl, to do the first step of its mechanism. Question 4, look at this reaction and draw coordinate diagrams. So this is just going back to what we talked about here. You have the, I got these arrows switched up. The tall peak is the uncatalyzed reaction. The small peak is the catalyzed reaction because you're lowering this activation energy to make this reaction speed up. All right, so the next mechanism we're going to look at is covalent catalysis. I can't speak right now. Covalent enzymes catalysis. There we go. So here we're taking acetoacetate, and the CO2 is coming off, and so your intermediate state is an enolate to make acetone. This is slow. It still actually happens in your body uncatalyzed, but it is slow. So let's see how we can make this faster. So what can happen is that you have a nucleophilic reaction between the catalyst. Your catalyst is RNH2 here. The R would be the rest of your catalyst. And what's going to happen is that you're going to form a covalent bond between your substrate and your catalyst. This is called an imine or a shift base. When this shift base is formed, you're going to speed up this CO2 leaving. So the CO2 is going to leave and you're going to form a double bond with this carbon. Then the reverse of our first reaction happens where an acid can come in and it will cause our NH2 group to eventually leave here. So the electrons break from this double bond, this carbon can take this hydrogen. Now we have a nitrogen that's positive. We don't really want nitrogen to be positive all that much, so it can leave as RNH2 with a base, and we are at acetone. So this is just how we're speeding up this reaction through covalent catalysis. So question 5, you can see that this bond is transient. That means it forms and it breaks. Why must covalent bond formation between the enzyme and the substrate be transient? That's because you want your enzyme to be functional after every reaction it does. So let's just say you never broke this enzyme substrate bond. Well, your enzyme could only do one reaction and it'll be broken, which would be a giant waste of energy. So let's look at some cofactors and their relation to mechanism. So let's look at metal enzymes or metalloenzymes, and 1/3 of all the enzymes we know of have a metal ion that it uses for a cofactor. And these metal ions help in 3 major ways. 1, they'll bind your substrate and orient them properly in relation to the other amino acids for your reaction to take place. 2, they help with redox reactions because the metal ion's oxidation state can change, so they can serve as the agent of change for the redox reaction in your substrate. You go from iron +3 to +2, and then at the very end go back to +3 again. 3, if you have negative charges in your transition state, your metal ions can help stabilize this. Remember, metals are positively charged, so they can help either bring negative groups together or stabilize a negative transition state. It can also help during acid-base catalysis because metal actually makes water more acidic. So it does that because the zinc will interact with the negatively charged or partial negative oxygen, making the hydrogens more likely to leave. And we do see this in the enzyme carbonic anhydrase. All right, so we talked a lot about enzymes, but what are the ways, why do we go faster? We know that it's because the transition state lowers, but how do enzymes actually lower the transition state? So one way is that they actually bring their substrates close together. So they bind the substrate, and if you have multiple substrates, binds both of them, kind of just shoves them that close together. Because substrates can't react unless they're close to each other. This is a proximity effect, and this speeds up a reaction roughly 5 times. Not only does it bring it together, but it orients the substrates in the correct manner to do their chemistry. Remember, these molecules have unique 3D shapes, and they have to be aligned correctly so they'll react. It's almost like Legos. Legos only snap, one way. You can't try and smash Legos together and expect them to automatically just snap together. So this is called an orientation effect, and it roughly speeds it up by 100-fold. Charge group. So during transition state, a lot of the times you'll have a charge group on an atom that doesn't necessarily want it. So your enzyme could help stabilize this charge group. Or this charge group may not want to form in water. And so the enzyme could expel water from the active site, allowing this charge group to form more easily. But the biggest speed up, 10 million or 10 billion fold, no, that is 10 million, don't know how to count, is that translational or rotational motions of the substrate in the catalytic group will stop. So inside any cell, any substrate, any molecule will be moving and it'll be rotating at the same time. Well, your enzyme more or less grabs your substrate and just make them freeze in place and then put them together so that reaction can happen. So that's the big thing. They are no longer free to move or rotate, which will speed up the reaction a lot. There is some penalties for it though. When you're holding them, you are decreasing a chaos, you're increasing order, so you do take an entropic penalty. But when you're binding your substrate and your enzyme, you're making new reactions, new interactions rather, and this free energy of binding can help overcome the entropic penalty. Now, when talking about enzymes want to bind the transition state. That's what the active site is really designed to favor. interacting with the transition state. And this goes back to the acid and base catalysis problem we looked at earlier on in this video. And we can understand that easily by thinking about this image over here. So we have an enzyme, and the enzyme's whole purpose is to break this metal bar. And our active sites are magnets. So let's say we bind the enzyme substrate, we bind the substrate really tight. So our energy diagram is our enzyme substrate makes a lot of good interactions, so the free energy of that's low, but our transition state is half broken or bent in this case. So it takes a lot of energy to get there because our active site is perfectly suited for our substrate. So this is actually slowing down our reaction, because if you remember, if the substrate's just by itself like right here, our catalyzed reaction is now actually slower. Well, if our enzyme is instead has magnets shaped like the transition state, what happens is that our activation energy, our double dagger, our enzyme substrate transition state product, for lack of a better word, is lower in energy. Therefore, it takes less energy to go over the transition state, and that goes to a faster reaction. And most drugs, antibiotics, agriculture, the way that we stop an enzyme from working is that we have our drug look like the intermediate state. So here we have a enzyme that goes from L-proline to D-proline. Here's the transition state. If we want to make that enzyme from stop working, we give it a substrate that looks a lot like the transition state, but a little different, so that our enzyme will bind it, but not be able to complete its chemistry. All right, question 6 is a review. What do metal ions do in catalysis? We talked about this. Interact with negative transition states to speed up the reaction or orient substrates. Basically, that slide over again. Question 7, why is it important for the enzyme to be compared to the reaction transition state rather than to the substrate? I just said that, so I'm not going to repeat myself, and it's in there in text as well. And that ends this video. If you have questions, please let me know, as we do have a test next week. Otherwise, I'll see you in the next video.